let's start with today's lesson. And what have we got here? We have got meiosis, and this is from junior. All right, and let's see what this question says. People, just remember, you are going to get questions on meiosis in paper 1 and in paper 2. You must know the different phases. You've got to know your interphase happens before everything. Then you've got prophase, metaphase, anaphase, telophase, and you have it for both. Okay, so here we go. The diagram below represents, let's get our navy, represents a cell during a certain phase of meiosis. Okay, so just quickly, when you have interphase, you can see nothing. And interphase happens before actual meiosis. So that's before. All right, then this is just when you have the DNA replicates. And we did replication last week. Okay, then you have prophase, metaphase, anaphase, and telophase. So it's PMAT. Prophase, metaphase, anaphase, and telophase. All right, now with prophase, you have crossing over in prophase 1, okay, and in prophase 2, there is no crossing over. Okay, I messed this one up. Let's just do this quickly. So if you look at prophase, you're going to see the cell, and you're going to see, there's your cell, you're going to see the nucleus, and you're going to see the chromosomes in their pairs, all nice. That is in prophase 1, and you're also going to see the crossing over. In prophase 2, you're going to see your chromosomes, okay, and they're going to have the little pieces that are already swapped. Okay, so you'll see that one, and you'll see another one, okay, etc. So that is what you will see. Then, when we look at metaphase, okay, during metaphase, everything is in the middle, like we can see here. So you're going to have your chromosomes here, lining up along the equator, either in 2 rows, okay, so 2 rows, then you know it is metaphase 1. And if they sit like this, your chromosome, 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 chromosome. So there is a single row, because those are going to all be joined. Then we need 4. Okay, then we're going to end up with metaphase 2, and there's a single row. Okay, then when we look at metaphase anaphase, anaphase is for away. And that's when the chromosomes start to pull from the equator to the poles. Here, you're going to have a whole chromosome pulling to the pole. Okay, that will be anaphase 1. Anaphase 2, you are going to have the chromosomes splitting, and they're going to pull to the... So here you've got whole chromosomes, here you've got at least a double chromosome, and here you've got your chromatids pulling through. And then telophase is easy. Telophase, you're going to have, you'll be able to see here that you've got 2, and here at the end of telophase, that's telophase 1. Telophase 2, you're going to have 4 structures. Easy, done. So those are the basics. You must know these basics. So if we look at our diagram, it says meiosis, and what are we looking at here? Well, clearly everything is in the middle, and if it's in the middle, it is metaphase. Okay, and something else is you can see your homologous chromosomes. Now, homologous chromosomes, you're going to have your 2 rows. So here's one homologous chromosome. Maybe that's the maternal one, and that's the paternal one. And here is the maternal one, and that is the paternal one, or the other way around. You've got your homologous chromosomes lined up. So there are 2 rows, and therefore it is going to be metaphase 1. If they were single chromosomes, then it would be metaphase 2. Okay, so it says, well, first let's label. So A and B, well, A and B are both chromatid, that they are non-daughter chromatids. And that's something else I need to quickly show you. Or how much space have I got? Let's just go down. You've got, there's a chromatid and a chromatid, and they are joined together by a centromere. And that's a chromatid and a chromatid. And those chromatids are going to be called daughter chromatids, okay? Because they are duplicates of each other, this one and that one, okay? Now, we'll have this chromosome here joined together. So there are 2 daughter chromatids as well. Chromatids. Okay, let's just extend this page a little bit more. Okay, also daughter chromatids, also centromere, and the 2 together make up homologous chromosomes. Okay, now people, homo means the same. Logos means position. Okay, logos, location. So it's the same position. They are chromosomes that are going to carry the same allele. So here, maybe that's the allele for eye color. Then we're going to have an allele for eye color on this side. So this will be the maternal, and this one will be the paternal chromosome. And the 2 together make up your homologous chromosomes. So they carry the same genetic or information, the same genetic information for the same characteristics. Okay, now we got that sorted. Then if we look at C and D, so C is, well, maternal, and this will be the paternal chromosome. Okay, and the 2 together, as I said, homologous chromosome. All right, so let's look at our questions. Let's identify the phase of meiosis represented by the diagram above. Well, we know it is metaphase, but which metaphase? Metaphase 1. Why? Because there's a double row of chromosomes. You've got your maternal and your paternal homologous chromosomes. Okay, well, next question says, give one visible reason, okay, for your answer, because we have, I'm going to just put the answer here, so let's do it here. And homologous, your answer for question 2 is going to be homologous chromosomes line up in 2 rows on the equator. Okay, and that's exactly what you can see. there's your maternal and your paternal and your maternal and your paternal, and you have your 2 rows. There's one, this here will be row 1, and this is row 2. All right. Then describe the process that caused the inheritance of a combination of both paternal characteristics on chromatids A and B. So if we look here, chromatids A, there, we've got that piece, and chromatid B, we've got that piece. So they've swapped from this side to that side and that side to that side. So what is the process? The process is called crossing over. So for question 3, we're looking at crossing over. Okay, so there are no marks for writing that. I'm saying that's what the process is so that you know what the process is. And this is what you would say. You'd say that number 1, or first point, homologous chromosomes line up next to each other. Okay, so we're going to have this one here, and this one here, and they line up next to each other. So can you see here, they're next to each other. All right, that's point number 1. Then point number 2, your non-sister chromatids. Now remember, what did I? These are sister, sorry, not daughter. They're sister chromatids, okay? And you've got that's a sister chromatid in this one, and these 2 are sister chromatids. So sister chromatid, sister chromatid, sister chromatid, sister chromatid. The chromatids from this one, the maternal chromosome, and the sister chromatids from the paternal chromosome, they are different. These 2 are therefore called non-daughter. I've got daughter on my brain. Non-sister chromatids, okay? Because they don't belong to the same chromosome. This one is a different chromosome to that one. Okay, so now let's go back here. So the non-sister chromatids touch at a point called and this is important, chiasma. Okay, so first they line up next to each other. Then they touch at a point called the chiasma. Okay, then crossing over takes place. Crossing over takes place when segments of DNA are swapped between the 2 chromatids. Okay, so you're going to have so this will be the maternal and paternal, doesn't matter which order they're in. Okay, they're then going to lie next to each other, and then they cross over. So this one is going to do this, this one's going to do that, and where they touch, that's your chiasma. And at that point is where they swap information, so that at the end of it, you're going to end up with, and that will be white, and this will look like that. And that will stay. So this piece here and this piece there, they are swapped. So what do we end up with? We end up with a maternal chromosome with a piece of paternal, and the paternal chromosome with a piece of maternal. And that, folks, is what causes variation. That is what gives us our genetic variation from one to the other, from one organism, at least from one individual to another individual. Okay, so we haven't finished yet. So crossing over takes place when the segments of DNA are swapped between the two chromosomes. So there and there, all right? And that results, let's do a capital R, results in a recombination, okay? Recombination, this one's got a piece of that one's got a piece of the maternal, results in a recombination, so again combining of genetic information or genetic material. Okay, and that's important. It's a recombination of genetic material on segments of the, and what are these? This one and that one, they are homologous chromosomes of the homologous chromosomes. Okay, which leads to genetic variation. And this point here, this one, you are going to get, you can be asked as part of a meiosis question, you can be asked as part of a DNA question, and you can be asked as part of an evolution question. So make sure you understand that. Crossing over, the chromosomes, you have the point where it touches is the chiasma, okay? Pieces of genetic information of DNA swaps between the two non-sister chromatids, so that when they finish, they recombine, and the paternal chromosome carries some of the maternal chromosome's DNA, and vice versa. So that each of those chromosomes are now going to carry part of each other. Because remember, only one chromosome is going to end, of the pair, is going to end up in a gamete. So whether it's a sperm cell or whether it's an ovum, only one is going to end up there. And it's the one that ends up there that is going to then be different, because they have now swapped information. So a child is never identical to either the mom or the dad. It's always a combination and bits and pieces of the other one. All right, now let's go back. I think that was the last part of that question. Oh, it says state 2 reasons why these chromosomes are homologous. Okay, now if we look, and I'm going to do this just on here. If you look at this, they are, so it's, how are they the same? Okay, that's what you must look at. And the thing is that chromosomes are the same if they are the same size and the same length and the same shape. Okay, and also the point of the centromere. And if all those things there are the same, the word same, if all of those are the same, then they are homologous chromosomes. Okay, while I was looking through here, I saw a question here that Mbali's put on. Just give me a sec here. Look here. Can you see these? Okay, these clearly are non-replicated chromosomes. Can you see? But this is what we call a karyotype. And every person on the planet has a karyotype. This is what you're going to see in the nucleus of every single cell in the body. Okay, so there's chromosome pair 1, 2, 3, 4, 5. That could be maternal and that's paternal. So maternal, paternal. So these are all homologous chromosomes. But if you look, these are longer. So chromosome pair 1 are the largest. And chromosome pair 22 are the smallest. Okay, they're always the smallest, and 1 is always the largest, and everything else is in between. But look here, look where the centromere is, centromere. But here the centromere is in the center. There the centromere is right at the top. And so on, there's another centromere there. These guys have the centromere pretty much in the center. So it's the position of the centromere, it's the length of how long they are, it's the shape. I mean, if you look at this shape, okay, look at this shape, and then look at this shape, it's a different shape. Or this one, those are different shapes. And if you, each time, it's a different shape. So they've got to be the same length, the same shape, okay, the same size. So I mean, like, these are big, these are tiny, 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 tiny little ones, okay, except for these two. Now, these two here are not homologous. Why? Because that's an X and that's a Y, because they are the gonosomes, or the sex chromosomes. All right, let's go back to our original question. And okay, so homologous, remember, homo, the same, logos, that they have the same pieces. So the same length, the same shape, the same size, the same centromere point. Okay, and look at these guys. These guys look the same. And if we go up a little bit, these guys look the same. All right, there we go.